And the reason is like, it's coming to this research, is like why we have 400 million people, around like 70% of them, they got hepatitis B virus. They never developed to cancer, while the other, they developed to become hepatocellular carcinoma and eventually death. So this is the hypothesis of my research. And as those million of people, they die eventually because of liver transplantation. And then after that, is like it's developed to the way they got autoimmune disease, and then the liver is going to get disrupted in a way that the people lose their life. So what's the most important in this research? We try to evaluate, is the immune system fail to respond to hepatocellular carcinoma, to hepatitis B virus, or the virus is very strong to the point that can deplete the whole thing. If the virus is strong, that means every research we have facing related to the virus, that means it's true. But how come the virus is very strong? If the virus is very strong, how come like 90% of people, they got hepatitis B virus, but they got cured? They got cured, they become fine, and those 90% of people, they never get hepatocellular carcinoma again. And when it's coming to the medical logic, that if we have 90% of people, they got hepatitis B virus, and then they got cured, then that means the virus is not strong. So what's the wrong with it? The wrong is related to immune system. And what's the important about the immune system is CD8 and natural killer cell. Because those two cells are responsible to kill any infected cell and any cancer cell. And why they fail to kill the cancer cell? Why they fail to kill the cell who got the virus? And the reason for that is related to the granzyme. And granzyme is one of the enzymes that kill infected cell. How it's going to kill infected cell? The CD8, as soon as they recognize this cell is infected with cancer or virus, it will recognize the cell, and then it's going to release the peripherin, which is a protein. This protein is going to make whole, and then the granzyme get in the infected cell, and then two things is going to happen. It's going to remove protein PCL2 from mitochondria, then all cytochrome C is going to be released from mitochondria, and guess what? The whole energy system is going to be depleted, and the cell is going to be lysis. Or it's going to initiate caspase 3, which is responsible for denaturation and breaking down the DNA. So why those two mechanisms, no one of them is working? How come if natural killer cell is not going to do the job, so CD8 should do that? Both of them, they fail to kill the infected cell and the cancer cell. And it's come to we try to evaluate why granzymes fail to do that. In physical chemistry, we have three things related to enzyme to work efficiently. First, they have to have substrate, they have to have low Km, it should have normal temperature, normal pH, but there's very important another factor, which is the concentration of the enzyme and the activity of enzyme. It's very important to kill the infected cell. And here we try to evaluate the immune system enzyme related to CD8 and natural killer cell. Here the diagram is explain everything related to my idea. I'm not going to talk about those receptors and how they kill, but here is the peripherin. The granzyme B is going to get in the infected cell or this cancer cell, and then the three pathways is going to happen. Either is going to go to PCL2, remove PCL2, and the cytochrome is going to be released from